Let's begin with 2024's paper. All right, we're gonna break it up into modules. So we're gonna start with module one first. All right. So question 1A1. List four properties of water which are important to life processes. All right. So let's start off. All right. What was the first one that we stated? Okay, high specific heat capacity. All right. And if you're looking at something like that, how does that relate? Why is this so huge? I don't like this. All right, but all right. So it speaks about stability. All right. What does the high specific heat capacity speak to? What does that mean? Okay. All right. So looking at stuff like that. So that's one property of water that may be important. Let's get another one. What's the next one? Latent or specific? Okay. So vaporization. So what does that speak to? I guess. Okay. So how would that be important to life processes, though? So you're speaking about. Go ahead. The blankets. Hmm. You mean the water vapor that exists in the atmosphere that acts as a greenhouse gas? Okay, lovely. All right, so you can mention that. Could also mention cooling mechanisms, right, for certain organisms, etc. Another thing. I'm pretty sure there are some in the chat, but I'm not doing the chat right now. But any anything else? It's a universal solvent. All right, so it's universal solvent. So how does that relate to anything? Mm-hmm. It does. Right. So it being a universal solvent, right? Majority of the biomolecules that are required for life, right, are polar in nature. Right. So water would be a universal solvent there. That means that majority, if not, there's a very strong if not all metabolic processes, right, occur in water. Water is the primary solvent that is required for all chemical processes that support life. Alright? So you could think of something like that. So even though it's not required to explain for the question, we need to understand everything. Right? We're not just answering question for question's sake, because this will not come tomorrow. I doubt the same exact question will come. But the same principle can come, so we must understand the principle. Alright, anything else? It acts as a repeat? Never, never quite heard that properly. Could you type that in the chat? A buffer. I guess it's a buffer, if you said. Okay. And we understand how buffers work because we do chemistry unit one. So, that's question. I have a very important question. I've lost track of time. Chemistry unit one, paper two, gone already, right? Okay, lovely. So let's move on. Describe how two properties of water are aided by the hydrogen bonding between water molecules. So it didn't say to use any specific property that we had above. Any other two properties are any property above or any other one. Density, surface tension, right? So we have these two. So explain the density of water now. What are the differences specifically? Hmm. That's an interesting way to phrase the answer. Um, but yes. Right. So ice is well, not ice. Water itself, H2O, is a miraculous molecule in that its solid is less dense than its liquid, right? So we know that due to the hydrogen bonding, right, within water, we're going to have, yeah, so we're gonna have like occasional hydrogen bonding, right, as water molecules move past each other. But as it starts to cool, it starts to space out and create a lattice structure, right? That allows that lattice structure to be less dense than if it were to be a liquid, right? That would allow it to be, as we said, less dense. Less dense things would float, so the ice would float to the top, and it can create insulating layers upon ponds, rivers, lakes, etc. If they are supposed to freeze over, right? And the insulation simply means it traps heat so that marine or aquatic life can thrive, so that we maintain ecosystem on the earth. That could be a thing. Reactivity in terms of hydrolysis, hydrogen bonding. Um, that could be another response there, right? Could be another response. As relates to surface tension, so we're talking about cohesion. Sure, cohesion and adhesion stuff. I mean, in this case, would be cohesion, right? So water molecules hydrogen bond with each other, right? And you could speak about how we can upscale that, right? So each water molecule will hydrogen bond with itself, with another water molecule, rather, right? So oxygen and hydrogen. Yeah, so one molecule, yes. So we're on point with that, right? So each water molecule can create four hydrogen bonds with other four hydrogen water molecules, right? So if we look at something like that, right, the sheer amount of water, for example, 1 mole of water molecules, etc., right? The amount of bonds in that specific area, right, would scale, well, the strength of that will actually scale up with the amount of water molecules. Right, so we're gonna end up with a certain tension, right, that tends to hold the surface of water, right, and that's basically what we call the surface tension there. Um, so you can explain it through cohesion via hydrogen bonding there, right? So there are many aspects of water that can be explained. Almost, not everything, but almost everything about water can be explained through the hydrogen bonding. All right. So I'm not here to write all the answers on the slide, by the way. We're discussing the answers. I might write one or two words under it so you can remember generally what our response was. Okay. And there are many other responses to the same questions. Right. So it's not that. So if you want to pose an answer, you can pose a viable answer. It shouldn't be a problem. Go ahead. You raise your hand. Mm-hmm. 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 You said that's semi-permeable membrane? Oh, hmm. All right. Okay. So lovely, 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 lovely. All right. So I'll accept that response, but let's attack the key terms and a few fallacies. Right. First of all, as I said, it's a simple transport, right? Remember, simple diffusion is a form of passive movement. Passive movement implies what? What does that imply? So no ATP needed. Lovely. That is what it implies, right? Another aspect of it we know is from high to low concentrations, right? So regions of high concentration to region of low concentration, right? What's another thing that we must state about simple diffusion is that it is down a concentration gradient. So not necessarily across a selectively permeable membrane, but down a concentration gradient, right? So the mere fact that it's passive movement means that it necessarily doesn't require ATP and generally would not really require a transport protein because if you require movement via transport protein, that transport protein would use energy, etc. Right? So by the fact that we know that it doesn't use ATP, it doesn't use a transport protein, right? So it doesn't need to necessarily travel through um, a membrane actively, right? But just down a concentration gradient from high to low concentrations, right? So we just need to mention generally these three things, right? And we should be able to explain our way to the answer, right? So it's 2 marks. We must mention there is a movement down a gradient, right? The passive movement of particles down a concentration gradient. Those are the two aspects that we require for these marks here. All right. Next response or next question. Figure 1 shows the movement of molecules across the cell membrane by three mechanisms. So I have X, Y, and Z. What are these three mechanisms? What is X? Hmm. Why are we saying active transfer? So it couldn't be facilitated diffusion? It couldn't be. Okay. But lovely. Right, so mm-hmm, mm-hmm. True. It's like that's like more into exploring plasmolysis and stuff like that, right? But having a look at this, right? So we know that if we have ATP and transport protein, that is active transport. But suppose we have Y now, we have a transport protein, but we do have ATP being involved. What's happening here? And look at the molecules that are being moved, rather than in Z when we have mostly water. So what are we looking at? Are we sure it's facilitated diffusion? Define. Okay, so we're looking we're looking at it down a concentration gradient, right? This is in Z here, right? Note now, guys, it's the first time I'm seeing this question. When I opened this paper, I went straight to the module 2 because people say we have issues with that, right? But looking at this, I think that transport protein is an aquaporin, right? Because I'm seeing water being moved, right? But it's being moved against its in its gradient. Is that true? Could that be a way we interpret it? Or does it? Okay, lovely. All right, so this would be facilitated. Um, Hopefully I can spell facilitated and don't flop. Lovely. No, they. shouldn't. Parent, the parenchyma? Well, eh, I reckon I can probably spell it. Alright, but let's have a look at this. Does it count as osmosis? Facilitated diffusion is the closest to osmosis, really and truly. Um, mhm, mhm. But let's have a look at this. So that's 3 marks there. This paper calm so far, right? Calm. There's nothing at all that is a problem here so far. 3. You are given the following apparatus and materials to identify the tonicity of each of 3 unknown solutions. First of all, what is tonicity? What does that mean? Yes. Yeah, but what? So tonicity refers to relative concentrations of solutes in solutions, right? That cannot pass through a membrane. Okay, but we're just looking at the tonicity, right? Hypotonic, hypertonic, isotonic, whatever. Those are examples of different states of tonicity, right? So we have all of these things. So red onion, access to water, um, if you live in a specific part of Jamaica, that would be a challenge. Blade or scalpel, distilled water, drop off forceps, all these type of things, right? What are we supposed to do now is really and truly tailor a PD, right? A plan and design, right? So plan and design are our structure, right? A procedure for investigating the tonicity of 3 unknown solutions. So how would we actually do that? What would be our procedure? And now, because it's a procedure, we can come up with multiple correct responses for this. So what would be a good procedure here? Mm-hmm. 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 So we have a control and we have the 3 different solutions set up. And what we're going to do is put the red onion in all 4. So prepare the slides, right? Because we have a drop on for and those type of things. So if we look at it like this, right? Based on the materials that we have, right? I'm guessing here we would prepare, would have the solutions, right? And would have distilled water, right? As our control. Would prepare all the slides and then use the dropper to add solutions onto the slides, and we will view them under a microscope, the light microscope, right? And then what would we be looking for? So we're looking at the turgidity of the cell, how it changes, right? We're looking at because it is a plant cell, right? We're going to be looking at, in some plant cells, you can look at the size of the vacuoles. In some plant cells, you can really look at the membrane, right? Whether or not the membrane is touching the cell wall, right? Or if it is a little bit encroached, a little bit more in, etc. Right? But that's basically what you're looking for. You're looking for the cell to react to the type of solution that you place there, right? And then you can now determine the tonicity of the three solutions based on how the cell reacts. So if the cell is more turgid, right, that would be what? Hypotonic? Are we sure? Would it be hypo or hyper? Hypotonic, right? And if it's going to be shriveled, right, or flaccid, it's gonna be hypertonic, right? And then we're gonna be looking at which one maintains the regular structure of the cell, right? Which that would just be isotonic, right? So you're just gonna have to plan and design that experiment there, right? So really and truly, it's from this question is about more or less common sense. So based off of the information that you know from the topic, you just use common sense and sit and you try to write your suitable procedure there. All right. Um, but more or less that is that. How many marks? 5 marks? Should be fine. 5 steps. 5 to 7 steps should be fine. And you have to basically you have to mention the control. You have to mention what we're looking for in the tonicity. You have to that basically. You have to mention the preparation of the slides, etc. How they carry out the experiment, as in using the dropper to drop the solution onto the thing, etc. It shouldn't be much of an issue. C1. Yeast membranes contain ergosterol and mammalian cells contain cholesterol. Figure 2 shows the structure of ergosterol and cholesterol. Alright, so we have these two things here. Really, really fine so far. Alright, okay. So based on the structure of ergosterol, right, compared with cholesterol, shown in figure 2, suggests the function of ergosterol in the cell membranes of yeast cells. Hmm? So one thing about biology, yes, most definitely, Jacob. Alright. So one thing about biology, there's one thing that you must note to never be able to fail biology. You must note structure and its relation to function. That's all biology is. You look at the structure of something and you relate it to the function. That's it. One, straight A profile, done. There's no question. Right? We must be able to do that. So what is the only difference here? Right? We look at that and we right? So the structure is more or less the same, right? More or less the same. In chemistry, of course, we'll state that these are definitely completely different molecules, right? But in biology, more or less the same, right? So what is the general function that we could suggest? As Jacob stated, it has something to do with the fluidity. hmm Another thing for biology, I beg you guys to answer in complete sentences or complete paragraphs. Alright? So we can create a complete sentence here. So since ergosterol, right, has a similar structure to cholesterol in mammalian cells, right, we can then assume that it has a similar function in yeast cells, right? Meaning that ergosterol would assist in the stabilization of cell membranes by regulating the fluidity, right? It is inserted, that would mean that it is inserted between phospholipids, reducing permeability to small molecules and maintaining membrane integrity under varying temperatures. We remember how cholesterol works? We should remember how it works in the cell membrane, in the fluid mosaic model. Yes. So what we're going to be doing is just relating the structure of the cholesterol. So really and truly, if you break down this question to bare bones, it's asking the candidate whether or not they understand what cholesterol does. That's really and truly what the question is asking. Alright? So after a while, you'll be able to just look at questions, right, in any topic whatsoever, any subject, and be able to break it down to the bare bones. Because a lot of times they just rant on and bust ears for some reason. Two, figure 3 shows three components. of the cell membrane of a red blood cell, right, our erythrocyte. One of the components forms the fluid bilayer. Another contributes to the blood group B, right? So we have that protein there. And one determines if an individual will be Rheus factor positive or negative. All right, so I was about to say an antigen presenting protein, but this is not unit 2. It's a glyco. It is a glycoprotein. All right, it's a part. Is it an intrinsic protein? Extrinsic rather. It's a transmembrane protein. It's a part of a transmembrane protein. All right. So it would present certain groups, right? Certain nucleic acids, right? That we can test to figure out whether or not an individual be risk factor positive or negative. All right. Risk factor. Wait, what did you just assume a while ago? Okay, what you just assumed something a while ago. No, the risk factor, right? So it's that extrinsic protein that was spoke about earlier, right? So it's a part of the blood group system that is crucial for blood compatibility, right? So it'll be able to tell you whether or not your blood is compatible with different stuff, basically, right? So being real positive, right, and being real negative would be important when deciding whether or not persons can. Okay, I don't want to get into. I'm going to be slowly going into Unit 2 biology, and I don't think that makes sense right now. Right? It speaks to development of antibodies and creation of antigen presentation on the protein, which we don't do in biology Unit 2. So let's just leave that there for now. All right. But we're asked to explain the functions of the three components, right? The cell membrane of the red blood cell is shown, right? So, based on the fluid mosaic model, you must present your response in complete paragraphs, right? So, what are some of the things that we see here? Mm-hmm. We see the phospholipid bilayer. Mm-hmm. Okay, so what type of protein would that be? Mm-hmm. It's not common. Yeah. So we just need to speak about three aspects, right? So we have three aspects here, right? The risk factor thing, right? Protein is really the some of the functions actually described in the paragraph above, right? So we just need to explain the functions of three other components of the cell membrane. So first of all, phospholipid bilayer. Can we explain the function of that? Can we explain the function of the phospholipid bilayer or phospholipids in general? Anyone? Mm-hmm. 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 Okay, so we can see based on the structure of the phospholipids, they will form a bilayer that separates the um interstitial space. Not interstitial space, by the way. Intercellular environment from the extracellular environment, right? That's generally primary purpose of it, first of all, right? For the glycoprotein here, the blood group is presented there, right? What would be a function of that? Explain a function of that component there. So there are three components of the blood cell here, the blood cell membrane here. So what's happening? So we know the function of the fluid bilayer. Let me check the chat to see any responses. Okay. Mm-hmm. So that is valid. That is valid. That is valid. All right. So glycoprotein is used in determining the blood type, right? And the risk factor is a type of form of cellular recognition, in a way, in a sense, right? So we could state those. Um. So we must note those examples there, right? Um. So we must note the phospholipid bilayer, right? So we must note the use of that. We have a glycoprotein, right? And we also have the risk factor protein there, right? So it's not a channel, you know. I don't believe that this is a channel. The risk factor, I'm not sure if it's a, well, it shouldn't be a channel. It would it would be an extrinsic protein, all right? So risk factor, right, is really an antigen, you know. So really and truly, it's an antigen. So hopefully the persons who are doing this paper understand that that this is an antigen, and red blood cells can be antigen presenting cells. Right, so that's the thing there. And we should know what antigens are from CSEC biology back in those days, right? So we must be able to explain the purpose of these three. All right. So let's not waste too much time. See that you have a lot of space to write about all. How many marks is this? Question mark. Hold on. Why is it still going? 9 marks to explain what? All right. So that means that you're required to bust ears. So 3 marks for each. All right. So the phospholipid bilayer, I'm pretty sure you guys can explain away 3 marks of the phospholipid bilayer, right? So speak about the structure of the phospholipid, right? Then we're gonna be speaking about the how it is arranged into the bilayer and what the bilayer does. That's more or less 3 marks, right? For the glycoprotein, right? We should know what a glycoprotein is, right? So it's a mixture of some polysaccharide, right, along with some of the amino acids, right, and it presents, right, something extrinsically on the outer surface of the bilayer, right, and allows for cell communication and recognition, right. So you're going to be speaking about those things for glycoproteins. Glycoproteins normally do those type of things, right. For the risk factor protein now, this will act as an antigen, right? So, do we know what an antigen is? We know what an antigen is? Nope. What do you mean no? Antigens? They're substances that trigger immune responses, right? But these are presented on the surface membrane of the cell, right? So, as we were saying about the risk factor thing, positive and negative, it speaks to the compatibility, right? Um, of blood itself, right? So usually things that, for example, if the blood cell, right, has a B blood group glycoprotein presented on it, right, and it has like positive risk factor, etc., right, or except it would determine whether or not these persons can basically allow blood of this type to enter their body for like blood transfusions, donations, and stuff like that. So it ties into that kind of biology, right? But really and truly, I'm not sure why you put risk factor here if it is not explicitly stated in syllabus. That students need to understand what these things are. But it's CAPE, right? So we will take it, right? So the same thing I was talking about to the chemistry unit 2 students about on their 2024 paper, or say a lot of stuff that are not explicitly stated in the syllabus. But everything shall be fine because the papers this year been moving soft from Cape Start. So hopefully biology will be soft as well. Alright.